This is a project that started based on a conversation that I had with Aaron, and he was asking us in an email, can you tell me the dark GPCRs that are related to wound healing? And we said, yes, you can do it. It's pretty easy, but this actually opens up, can we do it for anything? Like if somebody asks us any biological term, any disease, can we give them a list of genes for that are either should be studied or already been studied in that disease? And this can be done with Generift. So Generift is basically a huge list of millions of associations between genes and PubMed IDs. So what you can do, you can use the PubMed API to submit a query to PubMed, and then get all of the PubMed IDs, and then convert those PubMed IDs to genes. And then you can get a ranked list of genes based on the number of PubMed IDs you have. One of the things that we found that Generift is kind of weird how they put in the data in that database. So there are like those jumps in the annotations, and they have a lot of missing stuff. So Alex who sits in the back over here, he wrote code to create an alternative resource to Generift, where he basically queried all of the human genes and got the PubMed IDs for them, and then did some cleanup. So now we have a bigger set alternative to Generift. We call it AutoRiff. And then together with Alex and Brian, that is not here, they created this search engine. And here you can come in with any term, and you can search PubMed and get those gene lists that are associated. And also what we have is the ability to predict the genes that are most correlated to those genes from that list. So just an example, if we put in Michael McManus, our favorite IDG CI as a search term, we get all of the genes that he published and how many times he published papers about those genes. So this is a scatter plot. Every point is a gene. The genes that he published the most is this Drosha, and he published 26 papers with Drosha. And the reason it's here at the bottom is because a lot of other people publish papers about Drosha. So the gene that is at the top over there is a more Michael McManus unique gene. So this is Michael McManus sort of like sphere of genes that he published. And you can do that with any author in general. And then if you scroll down in Geneshot, if you can try it now, the app is already working. You get two tables. On the left is the results from the papers that Michael, like 26 papers, and then a fraction of all papers that were published for this gene. And then on the right is the prediction of the genes that he should be studying. So there are two tables here. One is based on co-expression, and this is based on our reprocessing of all of the geo RNA-seq data and creating a co-expression matrix between all genes, and then looking at these genes, how much they co-express with other genes. So this is the second tab. The first tab is based on a co-occurrence based on enricher queries. So we have the search engine for gene sets that is pretty famous. And then what we do with when people come to that search engine, we save those lists. And we don't publish those lists, we don't make them available, but what we can do with that, we create this co-occurrence matrix of genes against all other genes, and then we can see how much genes correlate across all of those submissions. So millions of submissions, we save them, and then we create this. And then we found that this is really, really good for predictions. Like you can predict function, and you can predict sort of like the gene in the neighborhood. So all of this actually has, all of this app has APIs. And what we would like to do, and what we think we can do in just one day, we can build a website that has the authors, that maybe have pages for each author, and then have other authors that are working on similar genes. So we can find the most similar authors. We can have the genes that they published already in the network, and then some other predicted genes and how those genes relate to each other, and what other authors, whether those other authors also studied those. So this is something that we can do in one day, and hopefully we can recruit some people to join our team